Welcome to this tutorial. The name of this tutorial is Structural Bioinformatics for Evolutionary Analysis. And we are going to try to bridge the gap between two large fields of computational biology. One is molecular evolution, and the other is structural bioinformatics. So the idea is that these two very large fields are connected. We usually know about these connections, but we are going to try to share with you the many places where the research in one field can help the research in other. And specifically, we want to understand how protein structure change throughout evolution. And to do that, we are going to hear from a small group of people, including me. My name is Claudia Alvarez. I'm a NASA postdoctoral fellow at Georgia Tech, and I study the early evolution of folded proteins. We are also going to hear from Carolina Sanchez. She is studying the early evolution of intrinsically disordered proteins. And finally, we are going to hear from Vyacheslav Tretyachenko, who is studying the effect of the amino acid repertoire on protein folding and function. So I hope you enjoy this, and we start with you, Carolina. Caro, you're muted. I think you're muted again. So, yes.
Now, can you hear me? No? Okay, thank you very much. Thank you, Claudia, and thank you to everyone for being here. So first, we will start talking about introduction to web tools for protein structure classification. The first part will consist of basic concepts, and the second part we will perform some activities. So we will talk about protein structure classification, but what is a protein? So proteins have 4 levels of organization. The first one is the primary structure, is the sequential arrangement of amino acids in a polypeptide chain. The second level is the secondary structure and is result of the interaction of the hydrogen bonds, and these form 2 stable structural elements called alpha helices, as the blue ones that you see here, and also another element that is called beta strands. And then we have the tertiary structure, that is the folding of the polypeptide chain. And then we have the quaternary structure, that is the join of 2 or more of these polypeptide chains into one single form that is biologically active. So now, how do proteins look like? If we take a look of many of the protein structures solved in the PDB, we see a wide variety of forms and complexes, but mainly we will see some globular or rounded shapes like these enzymes. But also we have another elongated forms of proteins, like this one. This blue one is the collagen. The collagen is located in the extracellular matrix and is in fact the most abundant protein in the human body. But so if we take a look into each of these proteins, we will see that they are composed of protein domains. And each protein domain is a structural, functional, and evolutionary protein unit in a protein. And in fact, for example, we are seeing here the ribosomal protein L7, and we see that this protein is composed of 2 domains. One domain, this blue one, has a globular shape, like the proteins that we see in the previous slide. But another protein has a more extended conformation. Okay, But we also know that these globular or three-dimensional domains of proteins can be classified in a hierarchical way. For this purpose, there are these databases that classify them. So I will start talking about CAT. CAT has 4 levels of classification of the domains. The first level is called class, and it's about the overall content of secondary structure of each protein domain. As you can see here, you have all alpha domains, but you can also have all beta or a mix of both secondary structures. The next level is the architecture, and it's about the three-dimensional arrangement of each of these secondary structure elements in space. Then we have the topology level, that is about how every of these secondary elements are arranged, are connected to each other. I mean the connectivity. But the last level is about homology, as this H says, and they are englobed in this superfamily level. So all homologous domains will be in the superfamily level. And the other very well-known database of classification of domains is SCOPE. It's very similar to the one of CAT, except that this SCOPE classification don't have this topology, don't take into account the connectivity of the domains. And there's a more recent database that is called ECOD, that instead of taking into account the fold and the topology, they use homologous relationships. So this, in fact, the superfamilies in both CAT and SCOPE have allowed analysis of the homologous relationships of all of them. And in fact, with deeper techniques of homologous analysis, we now know that many of the superfamilies that were previously classified as unrelated or non-homologous, we now know that they are connected. And these have formed a galaxy of folds. But we have a galaxy of folds in the protein universe. But if we see to the universe, we see that galaxies are formed of stars, of dust, but also they have dark matter. They are composed of dark matter that remain unseen. But we are talking about the galaxy of folds. And in fact, in biology, there are also some proteins that form part of the dark matter. And these proteins have remained unseen by traditional structural biology methods. But these macromolecules conform the dark proteome of the protein universe on Earth. And now we will talk about what are intrinsically disordered proteins. Intrinsically disordered proteins can be seen like a random coil, or they can have this shape of a spaghetti, but there are proteins that they don't have a unique three-dimensional structure in their functional states. They don't have to be globular to function. And in fact, these disordered proteins can exist as an heterogeneous, highly dynamic set of conformers, and they are highly flexible. As you can see in this image, they can be like a random coil, but then can change in some cases in fast ways to this pre-molten globule state that involves a random coil, but with some elements of secondary structure, and then to a molten globule, and then to ordered. So here we see a disordered protein of a cow. You can see that these disordered proteins don't have a unique three-dimensional state, but they are still functional, and you can see how highly heterodynamic they are. But as they form part of the dark proteome of the galaxy of folds of the protein universe, they are not classified hierarchically as the globular domains that we know. But SCOPE has a small part of classification of these IDPs or disordered proteins. But we also have some other resources to analyze them. We have a lot of, nowadays they exist a lot of protein disorder databases. The most common one is DISPROT. This DISPROT includes experimental characterization and functions of these disorder proteins. IDEAL is also a very well-known database. And I want to talk about MobiDB and D2P2 because this, besides including experimental characterization of these disorder proteins, also stores disorder predictions. So, but where is the place where all the protein solved structures are deposited? There's only one place around the world that deposits and is the unique repository of structural data is the Protein Data Bank, or PDB. And this PDB stores all the macromolecules like nucleic acids, complexes of protein and nucleic acids, and proteins. And it stores each macromolecule information in coordinate files that list the atoms in each structure and list their 3D location in space. But how did it all started? In the 1960s, Sir John Kendrew and his co-workers solved the first atomic structure of a protein. It was the myoglobin, and this protein stores oxygen in muscle cells. As you can see here in this image, this model of the first structure, the myoglobin, was very big and was composed of a brass model. And here we see Sir John Kendrew that, as you can see, in the 1960s, scientists used ties for doing scientific research. But so in the first model of protein sex structures, we have brass models, but nowadays you can download the atomic structures of any protein in your computer, because now the PDB stores the atomic data in coordinate file formats. There are two coordinate file formats in the PDB. One is the PDB format, and the other one is the CIF format. This contains atomic level data structure. So for example, you have one protein here. This is the PDB format of one protein. And you can see the amino acids. You can see the elements of which is composed, like this carbon alpha, and the coordinates in the Cartesian plane. You also have the chain, and you have the sequence number. These two formats are very similar, but it's very important to say that the CIF format can store more data than the PDB format. And in fact, in the future, the PDB format will be outdated and only the CIF will be maintained. But what information does the PDB format contains? Okay, the information it contains is about description of the molecule, also about author's information, also about the sequence. This is the sequence of the protein that was determined, but also sometimes in some X-ray structures that conform the vast majority of the PDB, they have sometimes missing residues that are located in the remark 475. And this is important to take into account because they were present in the sequence, but they can't be seen. But also you have this heteroatom record that stores information about some small molecules like these irons. But if we're analyzing structures, how can we measure the detail of the structural data? Resolution is one of the most common measures of the detail of the data. And in fact, what resolution talks about is how easy it is to see every atom in the electron density map. The lower the level of the value of the resolution in angstroms, the better, the easier you will see every atom in the electron density map. So you can say that if you have a structure of 1 angstrom to 1.8, it's a very high resolution structure. So if you are working, it's better to choose those structures with lower levels with lower values of resolution. But we have another structure, we have another way to say that our structure is with high quality. These are called the validation reports. And if in every page of the PDB, you will see this chart. We can talk about all of these values, but the most important are the R values. So we recommend you to see the R values. The R value measures how well the simulated diffraction pattern matches the experimentally observed diffraction pattern. As another tip, we can tell you that it's good that you see if the R value is similar to the R free. So this is all. So now we will go to the page to perform the exercise, the introduction exercise, and later we can answer some doubts and questions that you have. Thank you. So please, I will ask you to go to the platform in GitHub, and please go to the introduction section. And now we will perform two exercises. The first exercise will consist of about protein data bank exploration, and the second exercise will be about how to download the coordinate files, not the BRASC model, but your coordinate files into your computer. So we will ask you please to do the exercise by yourself, because in this way it's easier to you. And if you finish earlier than in 20 minutes, you can go and take some coffee and then return quickly, please. So if you have any doubts, please, you can ask us. So if you start doing the exercise, it will be great, please.
So while you do the exercises, we will be reading some of your questions and we'll see if we can answer them. Also, if you have any question on how to do the exercises, or if a link is broken or something like that, just let us know. And we'll come back to another presentation in 10 minutes.
Yeah, it's in 15.
In 15 minutes. Caro, you have one question. I
don't see the...
Ebru is asking how much of the protein galaxy consists of the disordered proteins?
It's more than 50%, but it was unexpected that recently some researchers found that a great part of this dark proteome, in fact, is conformed by orphan proteins, proteins that they do not have any homologous relationships with any other known sequence. So it's not only about disorder, sorry, it was in the slide, but probably I didn't mention it. I can send you the reference if you are interested in that. I will send you the reference now.
Thank you, Carolina.
Thank you for asking also. So, how can I send the...
So we can probably include it in the GitHub page we have set up for this tutorial, but also maybe in the chat, in the Q&A.
Yeah, I don't see the chat.
Maybe I think it would be probably better if
I will put it in the GitHub now.
All right. Yeah, I think so. Yeah.
Yeah, thank you.
I'm going to share my screen just to show you at the same time that you are following this tutorial. Maybe for those of you that won't be following it, I'm just going to put my screen on the GitHub page so that you can see what is the result we are expecting from this first part. It's actually this first exercise is just an exploration of the resources and the data. So, no. And we have another 5 minutes for this exercise. If you have other questions for Carolina, please send them. We'll try to answer them. If you are just joining now, this session is live. We are just working on the exercises in GitHub. That's why it is so silent. We'll be back in 5 minutes.
So yeah, while you are doing, guys, the exercises, we want to tell you something very important because, in fact, the PDB has 3 main repositories. One is the PDB of Japan, one is the PDB of Europe, and the other one is the PDB from America. So, but we know that the most complete in terms of information is the PDB of Europe. That's why we assigned the tutorial exercise to be doing to perform it in the PDB of Europe. And in fact, if you see in the further readings, you will see that that in the last, in the references, we see that we send you the link of the PDB of Europe. So I know that you are doing your exercises, but I will share my screen just to take a look together into this. And yeah, so I don't know if you already ended, probably yes, but I will take a fast look to this first exercise that is about the PDB exploration. So first you search for this emerythrin in the PDB of Europe, then you will play search, and then you can have a more detailed or refined search of this emerythrin. You can search it for even the taxonomic distribution of this, like this organism superkingdom. You can also search it for experimental methods. Now that we chose the organism superkingdom of Archaea, then we get into this structure that is of an Archaea of Pyrococcus. Then you went into the main page of this protein, and in this part, the gray box, you make a click in this molecule details section, and then you enter to this marvelous page that PDBE provides. That is, it includes information about the UniProt code, but also the FASTA sequence, but also you can see in this visualizer how is the three-dimensional structure of your protein. And in fact, you can see the architecture in terms of PFAM domains. You can also see the CAT, the structural classification domains database, to which domain it corresponds our domain. And very important, if you want to see the sequence, each residue by residue, you can click in this expanded button. And also, because this first part of the tutorial was about the classification of protein domains, so we want to know where are they located, classified in the CAT or SCOP databases. So for this purpose, we also went into the main page of the PDB of this structure. We also click in the structure analysis part, and we obtained, that is here, and we obtained this information. This is the biologically active form of the protein. In this case, it's a homodimer, and you also have more biological information of this macromolecule, like the chain, the length. Again, the source is an Archaea. But more important that we are interested about in this first part of the course is about what is the code in CAT, in the CAT database. And so yeah, you have also, we saw our structure in the PDB page, but there are many other visualization programs like PyMod. Here we added a list of many of these programs. And the second exercise consisted in retrieving your structural data, your coordinate files from the PDB. This is our goal. This is the goal. So you went into the main page of the structure. You found the upper right box in the quick links section. You went into downloads and click on the PDB file, blue link. So when you click in the download PDB file, you will obtain this structure that will be stored immediately in your computer. Please, if you have any doubts, please tell us. And at the end, we will also provide you our emails. If you have more questions, don't hesitate to ask us anything, please.
Okay, so now we are going to go for the second block. I saw a question from Alejandra who asks, how can we solve the ambiguity when there is more than one interpro ID for a protein when searching by name? And we are going to solve this question actually in this block of exercises. So in a few minutes, you will have an answer for that. So this second part of the tutorial is about searching for homologous protein structures. Right now we looked by name in the PDB-E, but we are going to cover first some basic concepts and then we are going to do some exercises. Probably you are asking, but why do we need to find homologs? And this is a very famous quote by Dobzhansky that says, nothing in biology makes sense except in the light of evolution. So that's probably the answer. But just to discuss a little bit more this idea, what we want to do when we study evolutionary analysis is we want to compare evolutionary units. And we want to tell a story about these units in terms of their relationships. We often use these schematic representations that are called phylogenies or phylogenetic trees to tell these hypotheses we take from our evolutionary units. So then what exactly is homology? And we have here a definition that says homology refers to a historical continuity in which morphological features are similar in pattern or form because they evolve from a corresponding structure in a common ancestor. So for example, we have in this figure the bones in the forelimbs of some vertebrates. And these bones are colored according to their correspondence in different species. So from this example, we learn that the classical definition of homology describes macroscopic features. And we also learn that these macroscopic features can have similar patterns or similar forms. And we learn also that we need to compare corresponding structures. But what if we didn't want to compare only things that belong to the macroscopic world? We probably want to classify all living beings. And this includes microscopic organisms. We can think, okay, well, why don't we compare the cells of macroscopic organisms and the cells of bacteria and archaea? And in this classical paper that was written by Carl Woese and George Fox, they discuss this. And they say that the eukaryotic cell cannot be directly compared to the prokaryotic cell. And they are referring to the cells as a whole. And they are saying this because according to the endosymbiotic theory of the origins of eukaryotes, we know that eukaryotes are a composite. So they are made from parts, and they argue that phylogenetic relationships cannot be reliably established in terms of non-comparable properties. So they say we cannot study cells as a whole if we want to classify all of the living beings. So we need a comparative approach, and we need comparable structures to establish phylogenetic relationships. So this is another classical paper that says and discusses why molecules can be our evolutionary units. And in here, Zuckerkandl and Pauling argue that in genes, mRNA and polypeptides, there is more history in the making and more history preserved than at any other single level of biological integration. I think this is very interesting, and this is actually what we use now many times to draw this hypothesis of relationship when we are using, when we are comparing very different distantly related organisms. So now we know that we can use molecules as traits that we are going to compare to establish these phylogenetic relationships. And in here I'm showing you one example of what you can do. So this is an example of the comparison of a molecule called myoglobin in different species. And we have these phylogenetic relationships in this diagram, and we have highlighted, this is an illustration by David Gotzel, and the colors denote the similarity or differences in the amino acid sequence of these proteins. And there's also something else that is highlighted here, and that's the heme group. And this is a molecule that is part of the protein, but is not part of the polypeptide chain, which also will store some information regarding the evolution of these molecules. And we can study that using structural bioinformatics. So now we have to talk about the types of homologs. We have two main types. One is orthologs, and orthology is the phylogenetic relationship where sequence diverges following speciation. So the history of the molecule will be the same as the history of the organism. So that's all that homology, orthology means. And just as a consequence of this definition, orthologous genes often have the same function, but this is not part of the definition. So this is a consequence, and it sometimes is the case, but we cannot know the function just knowing they are orthologs. We require other types of analysis to know the function of a protein. The other type of homolog are paralogs, and paralogs are genes that got duplicated, and they originate by duplication, and multiple homologs in the same genome will always or almost always be paralogs. And a very interesting characteristic of paralogs is that because they evolve by duplication, we can assume that there was an ancestral gene from which these two copies evolved. So when we look at paralogs, we can study things that happened before speciation. So just to recapitulate, if we follow the history of histones, for example, and if we go just to the red line, we have an ortholog. The ortholog would be histone H1, which can be present in humans and chimpanzees. But this histone H1 got duplicated at some point before the divergence of humans and chimpanzees. And now we have a paralog, and each of these separately will tell the history of the organism, but within a single organism, they will be paralleles. Now, what about structural homology? And we will probably find this term, or we will probably hear someone saying that they have found a structural homologue. And this is a little bit confusing because, first of all, homology is a qualitative trait. So it can only be homologous or not homologous. We don't need to qualify the degree of homology. So we actually don't say, I have a gene that is 25% homologous. What this gene can be is 25% similar or identical, but homology is not a continuous. It's a yes or no trait. We also don't say these two proteins are less homologous or are highly homologous. So think about this. Homology is like asking, are these two things relatives? It's just that we use it for evolutionary units and not people. So the question is just if they are relatives or if they are homologs, not how far away this relationship is. Another reason why the term structural homology is not very clear is because when you find, it's probably referring to something that is structurally very similar, but just by finding structurally similar proteins, it doesn't mean they are homologs. And this has been shown in this paper, for example, where the title is Structural Analogous Proteins Do Exist. And in this protein, we have here a random protein that was selected for ATP binding. And it ended up having a very similar motif to a naturally occurring protein. So this experiment of protein selection produced a very similar fold as a natural protein, and they are not homologs. So structure similarity alone cannot be used alone to infer homology. What can we use to infer homology? We need to use evidence from many different places. For example, significant sequence similarity, and then if we add to that similarity in structure, we will have a more strong hypothesis. And then if we have a similar function, this will be really strong hypothesis for homology. So for homology search, we usually use sequence-based approaches, and they can be sequence versus sequence, sequence versus profile, or profile versus profile. And if you are here, you probably ask, okay, but I'm interested in using protein structure to uncover these relationships. And yes, structure similarity can be a more sensitive probe for distant homology. That's why what we can do is, once you have a structure that is similar to one that you are interested in looking for, go back to sequence analysis and check if they in fact look as possible homologs. So right now it's time to try to do a homologous search. Let's go to the GitHub page and let's follow the second exercise of this tutorial. So again, okay, let me also share my screen this time. And if you go to the GitHub page, how do I do? Here. If you go to the GitHub page, the second exercise should look something like these. And we have two small exercises that you can follow. For most of these exercises, we have pre-computed the searches in case your search appears not to be working, or just not to saturate the servers that are going to be handling our search. So the first exercise is going to be about searching for homologs using annotation. And this is where the question about how to filter our searches when we have more than one PDB structure can be answered. So follow this exercise, and in a few minutes, I am going to tell you a very cool feature that was described, that was put there for the PDB-E to try to solve this question. So we'll have 10 minutes for this exercise. In 5 minutes, I'm going to discuss a little bit the first exercise. Okay, so maybe you are already in this part where you have seen the UniProt code for proteins. Sometimes you have repeated UniProt code, codes for different PDB entries. So if you click on the PDB-ECCB link, you will go to a page that looks something like this. And in this page, you can see all other proteins that have been crystallized if they correspond to the exact same protein in UniProt. A very cool thing that you can do is just visualize the superimposition of these structures, and they sometimes form clusters. So because proteins move, as Carolina showed us before, some of these are crystallized in different conformations. So if you have that with your protein, you will be able to see it in the PDB-ECCB. You can also visualize ligands or other similar proteins. Now, if you want to select one of these PDBs, what we would advise is first, if you are interested in a specific feature about your protein, you can check if, for example, it's a ligand that you are interested in. You can go to the PDB-E, check the electron density map and see if the quality of the model in the part that you want to study is good. If the quality is good, then go to the other parameters for quality and decide based on the parameters for quality for your protein, if you have the choice. Now, the second way in which we can search for homologs is just using sequence similarity. And for this second exercise, we are going to use the HHPRED server. We could use many other sequence-based tools, but this one allowed us to record our pre-computed calculation of similarity in this database, so it's easier to just load the result and then analyze what we got. So in the last section of this exercise, what I'm asking you is to compare what you obtain using these two different approaches. One is by name of the molecule, and the other one is by sequence similarity, and see if we get something interesting. All right. Maybe you have finished already. We could go to the next section in 5 minutes. let us know in the platform so that we can go to the next segment. There's a poll in the platform. All right, it seems like we can use a little bit of time, 4 minutes, and we change the activity. All right, 2 more minutes. If you have questions, please let us know. So there is a question about how to select a protein for function. Would it be better to use a knowledge-based approach or a similarity approach? So actually, the CAP database has a very cool page that is called Fun Families. And in this page, you can search for a specific protein domain family, and they have classified these protein domains by similarity in specific signatures that could imply that these proteins may have the same function. So as you know, sequence similarity can tell you a little bit about the function of the protein, but it's not always the case. So I would try to look in these classification databases. Ontologies are also useful, but these fun seem to be a very good tool if you are looking for a particular function in maybe a homologous protein would work better, or maybe you need to check if there are mutations that are making this protein not to work. So, and how to be sure if a sequence this is a very good question. So what if we have some sequence similarity, but the structures are different? So right now we have dealt only with sequence proteins that look similar and have similar structure. But there are a few proteins that either can change folds when they bind the substrate or when they do specific function. And there are also proteins that with just a few mutations have a totally different fold. These are very cool proteins, I think. And what I would suggest is to look for as many homologs as you can using a sequence similarity approach, and then trying to map the similarities of these sequences using a cluster analysis, because you can use a property of homologs that we learned in school, that is transitivity. And this is the property when the friend of your friend is my friend. So it means that if you find enough evidence that many sequences in the neighborhood, in the structural neighborhood of your two examples are homologs, then you have a strong argument to say these two different folds are in fact homologs. I think that's, so we didn't include those type of analysis in this first tutorial because we wanted to make it a very beginner level tutorial, but we can, if you want, you can send us an email and we can try to see your particular example. And yes, we are working on getting our slides for you. They were too heavy to put in the GitHub page. We are going to probably have to convert them into PDF and have them in the GitHub in the future, but right now we don't have the slides, we only have the tutorial. All right, so I hope you had enough time to finish the exercise. And I think it's time to go for the next part. You'll have the GitHub page and that will continue open. So if you want to finish the exercises at some other time, we can do that. So the next section is structure superimposition. Is that?
Yeah, the next section is called Overview of Pairwise Structure Superimposition Tools.
Thanks. Okay, this is supposed to be a short section, but let's see how it goes. We are going to review some concepts on the algorithms, and we just want to let you know what these scores mean, and then we are going to do an exercise. So I know this section is about the algorithms, but you have to remember that even though the algorithm is important, what is more important is the structural alignment itself. So it doesn't matter how fancy is the algorithm. the goal is to have the best superimposition because you are going to derive some scores from it. So always check your superimpositions. It doesn't matter which program made it. Okay, so with that, we can learn what is macromolecular structural alignment. And these are, the goal of a structural alignment is to obtain an optimal superimposition between two given protein structures. And the real goal is to have a large number of residues that are matching with a very low distance. And the distance is going to be evaluated using the root mean square deviation of the distance of two atoms. And we are going to, the algorithms usually perform rigid body transformations. So they treat your protein as a rigid body and they move in three-dimensional coordinates, doing some movements that are allowed. So one of them is translation and the other one is rotation. So after they compute translation and rotation, you will have your superimposition. So one of the things that the algorithm has to take into account is how to represent the data. And so this is the representation, a cartoon of a protein structure, but you can take, for example, the carbon alpha atoms as just points or spheres in space. And then if you have another set of spheres, independent spheres in space, try to locate the ones that look similar and compute a superimposition. That's the way these two algorithms work, DALI-Lite and CLICK. DALI-Lite actually takes a matrix of distances of each of the structures and looks for the similarity in these distance matrices. CLICK, on the other hand, takes these spheres in space and then clusters or makes cliques of these spheres and then compares these groups in two different proteins and tries to put them together. Another way in which we can represent our structure is as alpha carbons, but this time as connected alpha carbons. So that when we have one alpha carbon and we try to superimpose it to another one, we know that the next one also will be superimposed in a sequential way, and then the next and the next. And then if we have many alpha carbon atoms that look similar, we will have our superimposition. And that's the way these other algorithms work: Mammoth, CE, TM-Align, FATCAT, FAST, and Rapido. Okay, we can do it. There's a third one. What if we took secondary structural element and then just try to see if just the elements look similar in two structures? So it would be easier. There are larger portions that we are comparing. And just as what happened with alpha carbon atoms, we can have them as dissociated cylinders in space, or we can have them as continuous in a sequence. So there are a few differences in these two approaches, but the idea is the same. And these are other programs that use this representation. And the last one, probably there are others, but this is just a summary, is use graph theory and just convert our protein structure into a graph and see the similarity, the similar graph between the two of them, and produce superimposition of the, well, to look at the graphs that are similar, and we produce a superimposition. This is the way that many classifiers work because it's very fast. So this is a visual summary of the different approaches. The details of how they work are interesting if you want to develop or tweak any of these algorithms, but if it's not the case, you just need to know what your algorithm is doing in general. So sequential versus non-sequential. So many algorithms allow only sequential alignment, and this means that if we have two pairs of atoms, both pairs are going to follow the same sequence. So, okay, so if I can't, so if, for example, you have this pair and this other pair, B follows A, and then B prime follows A prime. So when that characteristic has to be true, then we know we have a sequential alignment algorithm. So to explain a little bit better, I think, this property of alignments, I have one activity for you. If you were able to bring paper clips, we are going to do an activity. Can you just let me know if you can all see my paper clips in the screen? Carolina or Slava?
Yep.
Can you see the?
Yeah, it's visible, but you have. I'm not sure if everybody can hear you, but yeah.
Am I okay? No, but you can see it, right?
Yeah.
All right. So let's imagine that if you can't see me, please let me know in the platform.
Yeah, I can.
Can someone please let me know? Okay. So imagine we have a protein that has this topology.
Claudia, wait a minute. Someone say that he doesn't see you, the paperclip. So I will
stop sharing your screen, maybe. And oh, I know. Wait just 1 second. I don't want to mess this up. Now, can you see me? Oh. Can you see me now?
Can someone say that? If we see, okay. Now they can see. Thank you very much for answering. Yes.
Yeah. And
yeah, thank you.
Is the sound okay? Do you hear me also?
Yeah.
Can you hear me?
Yeah, I think we can hear you. Definitely.
Yes, okay. Well, I'm going to assume everyone can hear this screen and hear me well, if nothing else happens. So, okay, we have one protein structure here that follows this topology, and let's assume it got duplicated, and then this is just a secondary structure representation of these two proteins. And the first one has a fold that looks like this. The small part of the paper clip will go towards us, and the large part will go to the back. So it will look like this. And we have another protein in which the fold changed. So the small part of the paper clip now goes to the back, and the large one is going towards us. So I'm going to put them side by side. to see that the connectivity hasn't changed. The only thing that changed is the way they fold. All right, so our goal is to superimpose these two homologs. We know they got duplicated, right? The first algorithm will take into account the linear connectivity of these elements. So what we are going to do is going to turn them, is going to take the small part where this protein begins and just align it like this. And we see, well, all of this is not superimposed. Maybe if I continue to superimpose my structures such that I get the most of the surface aligned, I will get something like this. So in the end, this algorithm will only be able to superimpose two helices. We think these are helices. The best one will superimpose the largest ones. So let's go back to how we started. We started with one protein that looked like this, and the other that looked like this. Okay, I didn't change anything, okay? Believe me. Now, this was the linear superimposition way. Now, what if we changed completely our philosophy and just don't care about the sequential way in which these proteins go? Just try to superimpose as much as we can. So I'm going to take my paper clips. I'm going to leave one immobile, and I'm going to turn completely the other one and just translate, do a translation in space. And this is the superimposition, but now we see that the sequential relationship is completely distorted. So the place where one of the elements starts is the place where the other ends. So this does not make sense in biological terms. So let's go back to the beginning. We have this again. And the third algorithm will allow some break in linearity, but we'll try to find some sequentiality, just not to break all the rules. And this algorithm will be just superimposed like this, such that the small part of one goes with the large part of the other, and vice versa. So this summarizes the three types of algorithms, or the philosophies of behind these algorithms. I hope you find this useful, and I'll go back to the presentation. Okay, am I back? All right. Oh, yes. So just to summarize this, what we saw using paper clips. In one algorithm, we will have only the two helices superimposed. This is a sequential topological algorithm. In the sequential but non-topological algorithm, we will have, oh, this is the last one we did in the exercise, in which we will have the small going to the large pieces of the first protein, and the large superimposing to the small pieces of the other protein. This is a sequential, but not a sequential with a threshold, and it's non-topological. And then the last one will not be biologically relevant, but it's able to superimpose the most area between the two paper clips or proteins. And this is a non-sequential, non-topological algorithm. So how do we measure the similarity between these two proteins? We are going to use, as we said before, RMSD. And this is the sum of the distances of the carbon alpha carbon atoms that we superimposed. And we are going to divide them by the number of atoms that were aligned. It's a quantitative measure of similarity, and it's expressed in length units, and the length units that we use for molecules is angstroms. But what happens if we have two proteins that look very similar, just the last part or some part of it is not as similar structurally? So this will completely bias our RMSD score. So RMSD scores are very sensitive to this type of things. So even if we have a structural core that is very similar, if we have many loops or many parts that move, the RMSD is going to be greatly affected, and it's also dependent on the length of the protein. So for that reason, other scores have been developed. And this score, for example, takes into account just the number of atoms that were aligned. And it also normalizes by the length of the target structure, such that it's not as sensitive as the RMSD on these very different regions of a protein. It takes more into account the parts that are similar. The TM score goes from 0 to 1, and 1 indicates a perfect match. And it's very good to use TM score. At least it's very easy to understand the meaning, because you will know that for every protein that you compare, if it's close to 1, it means they are very similar. There's another score, and this one is also a normalization of these very non-similar regions, specifically everything that is further away from 4 angstroms after superimposition is not taken into account, and it also factors the size of the protein to try to avoid this dependency on the length. So now you know more or less what each superimposition algorithm is able to do, and let's go to the exercises for this part of the tutorial. So this part, it's a bit heavy on the exercises, so I hope you have time to follow them. We will ask again in a poll if you finished.
Hello to everyone again. We also started another poll about, you can please tell us if you are finishing, you are close, or you haven't finished at all. So please tell us so we can wait for you. And those ones that finish earlier, you can go for a coffee or you can go to the toilet or something. We will wait for you, but don't don't be late too much, please. Thank you.
Remember that there are some links with the pre-computed results so that you can open them in another window if you feel that some of this is taking too long. There are a lot of steps in this one, so don't worry, we have time. And if you have questions, again, just we'll try to answer all of them if we can. You may also notice that we have a bonus exercise in here. This is another type of superimposition that we didn't describe, but it takes into account the metal binding site. So if you want to check the bonus exercise, you are welcome to. But if you feel like you won't have time, just save it for later. I think it will give you ideas of how to improve your own structure alignments and how to check for the quality of the ones you already did. In my experience, it's always good to use the visualization tools that these web pages have, just to give you an idea of what is happening with your superimposition, and just select the one that works best for your own protein. This is of course the case only when you have a number of proteins that you can manage to see one by one. So the third exercise is not necessary, don't worry about it. You can check it later. We have 4 more minutes to finish this part. But if you finished, feel free to take 4 minutes break. We'll be back soon.
we have more questions. We will answer them for sure in the page, but also here. So I will read the question about, hi, I've submitted 2 structures of my choice in Click and TM Align. Click gives me an RMSD of 1.71 and TM Align a bigger one of 2.94. So is Click better than TM Align? I may have missed it during your slide presentation.
It's a good question. So the RMSD, which is the score we are comparing in this particular example, will measure the sum of distances, the average sum of distances between the 2 structures. So a larger distance means less well aligned. In this case, it seems like Click did a better job than TM Align. And another thing to take into account is not the RMSD alone, but with the number of residues that the 2 superimpositions got. So if we have a smaller number of residues aligned with a better RMSD, it may indicate that it only aligned a very small fraction of the protein. But I am going to admit that I like Click a lot. It's a very slow algorithm, but it usually does the work very well. Just be careful and always try 2 alignment algorithms and check the structures. So use these applets to look around and turn around your structure and see if you can see what made the difference, which part of the protein it misaligned, or if one of them looks really better than the other. But yeah, RMSD, the larger the RMSD, the worse the alignment is.
Okay, I will go with another question. So, so Maria Morales asked, is there any program that permits us to do this superimposition analysis with multiple proteins at the same time?
Yes, and we are going to use one of them in the last part of this tutorial. But if you see this GitHub page, I included a link to Wikipedia. And in Wikipedia, you can find a very large list of structural alignment programs. We can probably go over it very quick. And one of the columns allows you to see if they can do pairwise or multi-protein alignments. The thing, well, I like doing multi-protein alignments, and usually these algorithms, what they do is they take one structure and then align the most similar of your set of proteins, and then sequentially add to the superimposition the rest of them. Of course, to do this calculation, they first compute a matrix of similarity, and then they take this matrix of similarity and do the sequential alignment of all of your structures. But yes, here you can find many programs that are able to do that. Let me just show you the ones that may have this feature. Well, actually, in this table also you can see if this feature is available in a web-based server, or you have to install the program in your computer. So this is a very good snapshot of what is available, although there are so many algorithms that it's hard to keep up with the piece. But the one that we are going to use is Matras. Should be somewhere here. We have 5 more minutes. We can, if there are more questions, we can answer them. But otherwise, you can take this 5 minutes break before the next session starts. Well, the next block.
Okay, so I will mention another. Okay, Diego Amaya Ramirez is asking if HHPRED is designed for working with heterodimer cases.
Well, HHPRED will do a sequence-based share. So you can only put one chain in this search, and you would have to retrieve the search for each element in your heterodimer. Yeah, so it's a sequence-based search. Actually, it's a profile-based search, and you have to look for each individual chain one at a time.
Great. The 70% of you have finished. I will mention about another question of Claudia Mirella. If I have a protein with disordered regions, these regions are difficult to find homologies. What method do you recommend to find homologous? Okay, I will talk again about disordered proteins in the next section, so we can answer to that question after that, Claudia. Thank you for asking. And okay. Shaila Verma asked if a protein does not has a structure and is modeled using available homologous structure, let's say with 50% sequence identity, still can the faults be predicted correctly by modeling? Yeah, we will also talk about in the next section about protein structure modeling. So yeah, so we are almost finishing. Everyone is almost finishing.
All right, so as we are approaching the next section, I hope you have time to do this exercise, and I just put a new question in the poll. So I don't give you an answer of which one is the best one, but I want to hear your input. Which protein structure superimposition did you find will give you more information about these particular molecules that we used in the example? So if you finished and you have a preference, answer that poll, please.
So the 80% of you have finished. So
yeah, it's
for the 20 or
I think,
or they can finish later.
Yeah, I think if the remaining 20% have particular questions you want to ask, just contact us after the tutorial. And as I said, the tutorial is going to be in GitHub for you, so you can go back at any time. But it's time for the next presentation. It's the turn of Carolina.
Okay, so I will start sharing my screen. So this section is about correspondences between protein sequence and structure. The first part will be about theoretical concepts, and the second one, we will perform a prediction of an structure based in a template-based way, and we will predict disorder regions in our protein of interest. So, in the 1960s, Christian Amundsen proposed that the unique three-dimensional structure of a protein is determined by the amino acid sequence. And if the amino acid sequence determines the structure of a protein, then the structure will perform a defined function. In this example, is an enzyme, the twin barrel, that you can see and you remember is a three-dimensional fold, is a well-folded protein. And this is part of the structure function paradigm. But what happens, this is not always the case. There are some also these disorder proteins that form part of the dark proteome of the protein universe that can function even without a well-folded structure, even without any globular structure. So this is part of what we call a disorder function paradigm, and is an emerging field. Okay, so, but we looked at sequence determines the structure, and also some sequences determine disorder. But can we predict the structure of some proteins if they are not any known structure? How can we do it? So, is it possible to predict protein structure? And the answer is yes. There are two main approaches: the template-free or de novo or ab initio approach. And there's another one called template-based or homology modeling. So the template-free consists of when you want to predict a protein structure or model a protein folding, but you do not have any similar or any homologous protein structure determined experimentally that are similar to your target sequence. So you go directly from the sequence, from the amino acid sequence, to the prediction of the tertiary structure of your protein of interest. This template-free approach is used by Rosetta, that is very famous, but more recently AlphaFold is another predictor that is, they are both very reliable. And also there's the other way to predict protein structure is a template-based or homology modeling. This template-based is based in the use of the similarity of your sequence, of your target sequence. Then you will search for all known experimentally determined structures in the PDB. And if one of these is similar to your target sequence target, you will model some structures and you will find a final possible structure. You will predict it. So let's talk about how this second approach works. The main steps will involve the multiple alignment of your target sequence. To first, the programs will take your sequence and then they will search for some sequence homologs. Then they will perform a multiple sequence alignment and the sequence of already determined structures. And if one of these is similar to your protein of target, they will call it template, or in this figure is called parent structure. This is the first step. The second step will involve the alignment of your target sequence with the template structure, the more similar structure to your target sequence. Then the next step involves the model refinement. It involves the analysis of some mutations and insertions, deletions present in this target sequence alignment. Then what will happen, this model refinement will produce a lot of models. And then after some sampling, you will have as a result, a selected, you will select, in fact, you can select it, a predicted model structure. This is very used, we can say. But what happens when you don't have any structural template, or you don't have any known structure that is similar to your sequence, to your target sequence? And the first step will be the same as the past one. The first step will consist of a construction of a multiple sequence alignment of the target protein with related sequences, with homologous sequences. The next step will consist in predicting local structure features, like secondary structure features. Then also using this multiple sequence alignment, and in fact, this is one of the most of the steps that give robustness to this prediction, is the prediction of residue contacts. So after that, you will have these predicted features, the local secondary structure and the residue contacts, will guide the model, the building of a lot of models. Then these lots of models, these are taken from Rosetta, will produce a refinement trajectory, and they will be refined and then selected. But okay, this seems a bit weird. So you have a sequence, then you want to predict the folding based on this sequence properties. But this is reliable. This is like probably true. How can we know it? So don't worry, anyone. That's why there's an experiment called CASP, Critical Assessment of Structure Prediction. And CASP, created in 1994, is a wide community experiment to determine and advance the state of the art in modeling protein structure from amino acid sequence. And I want to go to these two structures. So CASP consists in giving to all the participants, these experiments, all of the participants have a target sequence. From this target sequence, they have to predict the structure only based in that. but they don't know that another structure is already solved. So as you can see, this is the most accurate Cas model of the last experiment. It was 2 years ago. And you can see that is so similar to the one determined with an X-ray diffraction. They are almost the same, except you can see in these red loops that they differ, but globally they are very, very similar. And in fact, this is a very rapidly emerging field. And in fact, one of the things that have made that this rapid boom and success is the use of deep learning techniques to predict the inter-residue distances. So the new methods essentially solve the long-standing problem of predicting the fold topology of monomeric proteins. Okay, so we talk about how to predict the folding of structured proteins. But how can we predict disorder? Is it achievable even to predict it? So, okay, intrinsic disorder proteins are, okay, that looks like random coil. They are not globular, but the distinct sequence features that are present in these disorder proteins and regions, in fact, allow the construction of sequence-based rules that can facilitate high performance disorder prediction. So the disorder proteins, we have here the 20 amino acids, the disorder promoting residues, the red ones. You can see that they are not very hydrophobic. They don't promote the formation of a hydrophobic core like globular proteins, but they are less hydrophobic, so they are hydrophilic and they are very flexible, in contrast with the order proteins that are hydrophobic and less flexible. But not only these properties are like this, these properties have led to the creation of disorder predictors. And as you may know, there are a lot of disorder predictors nowadays. So, but how can disorder is predicted? There are several strategies of intrinsic disorder prediction. One of the most successful ones consists of meta-predictors, that is the consensus or the combination of several disorder predictor methods, and yeah, these have resulted in increases in the prediction accuracy. For example, we have here in this graph a list of many disorder predictors, and as you can see, this part, meta-predictors, are one of the most accurate ones in the prediction, these blue bars. And another strategy of predict intrinsic disorder is with machine learning. And these predictors employ some training sets for machine learning. For instance, some missing residues in the X-ray structures or some matrices of profiles of homologous sequences. So some of the most, if you can see here, for example, the disorder predictor more accurate is spot this, that employs evolutionary information and use also deep learning techniques like the most accurate template-free predictors. So, but we also, and in fact, the first ones were only based, the first disorder predictors were based only in sequences properties. And they, for example, UPRED, that is one of the most common one, and is very fast. You can analyze with UPRED complete proteomes or proteins, individual proteins. Is based because sequences with lower interaction energies are considered more likely to be disorder. Okay. But in fact, how common is disorder in biology? In the beginning, the scientists thought that there were maybe some exceptions, but these disorder predictors were pivotal for the establishment of the intrinsically disordered proteins field. Why? Because in the beginning, people thought like, oh, maybe my protein have a region that I can see in my X-ray diffraction atom records, but maybe it's not functional. No, but then later on, they realized that they were functional, but indeed, with the analysis of complete proteomes with the disorder predictors, they realized that in fact, the 30% of the human proteome is composed of intrinsically disordered proteins and regions. Not only that, if you see this chart, it's a chart about the prediction of disorder residues. And okay, these blue dots corresponds to viruses that they are biological entities, but they are not alive. And also you have eukaryotes, but also disorder is present in all cellular lineages or all domains of life, like bacteria, archaea, and eukarya. So bioinformatics was pivotal to the establishment of this field, and now we know that the proteome of all living beings and non-living ones, like viruses, are composed of structured proteins or globular proteins or ordered proteins, and also of intrinsically disordered proteins. But we also know, and we want to emphasize that because some proteins are composed of domains, some proteins are composed of globular domains and also disordered domains. But don't worry, the CASP in the past have evaluated also the disorder predictors, but since 2 years ago, the disorder community has I think that it's a very good idea, and inspired by the CASP experiment, they started to create this new experiment called CAIT, critical assessment of intrinsic protein disorder. And this is a wide experiment, and this also determine the state of the art in the detection of intrinsically disordered residues from the amino acid sequence. And what I really want to point you out in this slide is, if you want to predict intrinsic disorder, I will recommend you very heavily to take a look into this site, into this experiment, because they will publish the results. And the first experiment led to these results. I will tell you that every one of these are a different disorder predictor. And this one of the gray bars are the 10 disorder predictors with the best accuracy ranking. So, and more importantly, is that if you can see here, they are almost the same accurate, but they differ in the time in what they predict. So for example, you have very fast disorder predictors like Spritz, the Disprot type. And also you have very more, less fast predictors. So we will recommend you really to see to this page. And we talk about these disorder predictors, and we want to point out that the emerging field of disorder proteins is very, very, very well determined. Although it is emerging, the merge of these computational tools in combination with experiments will provide a better picture of this star proteome. But now, in this context, I want to ask for a presentation of a new bioinformatics tools to analyze these disorder predictions, this disorder analysis. So I will let Slava to talk about a new bioinformatic tool that allows us to analyze disorder in proteins.
Slava, I think you are muted.
Yeah, let's wait please for a second.
Or there is a small problem in the...
Yeah, there's a technical problem with, we will...
Still nothing.
Attention here.
I think now we are...
Yeah.
So maybe while we wait, we can go back to the question that was asked, and maybe Slava, you can interrupt us when you have sound. We will let you know that we hear you. But there was a question. So can you find homologs in intrinsically disordered proteins and regions, or do you lose all evolutionary information in these regions? How would you answer that, Carolina?
Okay. Sorry. Okay.
Caro?
Yeah. Sorry. Yeah, I was talking with Slava because there was a...
We can let you know when we hear you, Slava. Or...
Yeah, just 1 second, please. Or meanwhile, we can answer another question.
Mm-hmm. Well, he gets...
Okay, okay, I will answer the one with the disorder regions. So it was this question, I'm seeing that this from Claudia Mirela Osorio Morgollon. So if I have a protein with disorder regions, these regions are difficult to find homologs. What method do you recommend to find homologs? This is a very good question. First of all, disorder proteins some of them are characterized because they differ in sequence a lot. So it's not very easy. The sequence is not very well conserved. And in fact, this is a very important feature because biologically, we can say that it's important because they can even recognize different partners and they can change the conformation very easily. But I will recommend you to search for disorder proteins, making maybe profiles and also comparing some putative homologous sequences and determine the protein domain architecture and see if they share the same architecture, if they belong to the same taxonomic range. But I will also answer you in the platform. I will write you the answer also. Thank you very much for asking.
Yes, and I see that you found a way to solve the issues. So while you prepare the presentation, we are going to have Slava talking about not only predicting disorder, but what if you wanted to make a new protein with the composition you wanted? And that's what he's going to talk about.
Hello, everybody. Thank you. I'm sorry for the technical problems with my computer. So I got my 5 minutes to talk about computer software developed in our laboratory. And what I wanted to tell that the properties of disordered proteins, apart from being of sequential character, are also largely coded in a specific amino acid composition that can be notably seen in low complexity domains, which are composed only from a few specific amino acids, or it can be seen in disordered proteins with the defined amino acid ratios, which are very different from the amino acid ratios found on the structural proteins. Low complexity domains are usually found in a phase separating intrinsically disordered proteins, and the chains with less degenerate sequences are usual for proteins which, for example, fold upon binding. So these are the two typical ways how you actually code the intrinsically disordered proteins. And this can be seen that the ratios of the disordered proteins are very different from the ratios found in a uniprot sequences or just a PDV set of ordered proteins. It can be seen on a panel B in the bottom. Disordered proteins are unusually enriched in proteins, glutamic acid, serine.
I think we are stuck in one slide. Can you reshare your screen? Put it in center.
Can you see something else?
I see a slide that says amino acid composition in protein disorder.
And do you see all 3 points there?
Yes, but
Yeah.
that's what we were showing.
Okay,
Or
okay.
maybe just make it.
So the point of this slide is that disordered proteins can be coded just by amino acid composition. So in our lab, we designed a tool which can design a DNA library which will code for several disordered proteins, the whole set. So one DNA library will code for several different proteins where amino acid ratios will be fixed, but the sequence would be shuffled. So that can give us the tool for actual experimental screening of potential proteins in the large DNA libraries. So the tool is called Collide. I think the slides are not changing.
Okay. It should. Sorry, if you are seeing the Collide, Claudia, you are seeing the Collide combinatory library design?
I think I am still in the amino acid composition in protein disorder.
Okay, that's interesting.
Maybe just stop sharing and reshare.
Okay. Yeah, thank you. You need to pay attention because this is a new trend in bioinformatics. So, okay, I will.
Yeah. Sorry for the technical difficulties.
Yeah, but wait. It's worthy. So come.
It will be.
Okay.
Ah, that's it. Yes.
Oh, can you see it now?
Now we see the other computer's screen.
Uh-huh.
So.
Oh yeah.
Yeah. It will be solved in a minute.
So as I was telling, that the amino acid composition plays a crucial role in a disorder. So we tried to design such a software which is able to allow us to design billions of different sequences with a fixed amino acid composition in the same time. The software works with the degenerate codons. That means that the input for prediction is just the amino acid ratios which you need in your protein, and also the length of the proteins, in the final library. And the output of the program is the degenerate codon string, which when synthesized will allow us to get the library of combinatorial, the combinatorial library of sequences, where each sequence will code for a specific protein with this fixed amino acid ratios and but the sequence is kind of shuffled. That allows us to screen the vast libraries and explore the sequential space of different kinds. For example, we can screen for protein disorder, and as well we can we can screen for phase separation properties or other properties connected with the amino acid composition of proteins. Not sure. Can I share the screen now?
Yes, Carolina will have to stop sharing the screen.
Oh yeah. Do you see the Collide software now? Do you see the Collide slide?
We see the Collide.
Yeah. So the software, the input for software are the amino acid ratios, which are seen in left, where you can easily put the.
Can you share again your screen, Slava? Sorry.
Ooh. Okay. Sharing it now.
Great.
Yeah, am I back with the Collide slide?
Yes.
Yeah, so the input for the program are the amino acid compositions, which we need in our final library, and program will try to approximate this exact amino acid composition to output the degenerate string, which will code for the library of disordered proteins. In our case, they are disordered, but you can code any possible feature which is coded by an amino acid composition. We are not going to show exactly the program here, but you can easily access it on the GitHub. The link is shared in the presentations. So thank you for attention. Sorry for technical problems. I'm passing the screen to Carolina.
Yeah, thank you very much for seeing this new trend in bioinformatics, in this emerging field. So please, we can continue to go to the GitHub page. And now we will perform some template-based structure prediction and also a disorder prediction. So please go, I'm sorry. Okay, so please go to the activity of sequence structure. So let's access to it together. Sequence structure activity. I hope that everyone is here now. And the dynamic is the same, please. We will give you some minutes to answer and to practice by yourself the exercise. And you can ask us for any questions. And the first exercise, we will be about the template-based structure prediction. And we, for this purpose, we will use this Swiss model site. So now we can access again to the platform, to the GitHub platform. And please go to the activity of correspondences between protein sequence and structure. That is also called sequence structure. So we will perform, as we say, 2 exercises. The first exercise is about template-based structure prediction. And the goal is to predict a protein structure by homology modeling based on the sequence similarity to another protein whose 3-dimensional structure is already determined. So please start to doing the exercise. Any question that you have, please tell us. As a reminder, we will answer you all of your questions in the platform. So don't worry if you miss something when we reply to you in these sessions. So please start and we will wait for you.
While they are doing this exercise, so do you remember this question that said, if you have, don't worry, I'm going to tell you the question. The question was, if you have a very low sequence similarity, or I think it was exactly 50% or so sequence identity, can you still predict or model your protein?
Of Shaila Berma, right? This question, protein does not have a structure.
Yes.
Is it available? Still can the false be predicted? Okay, I need to think about this question. Right. So if a protein does not have a structure, and it's okay. So we're talking about some template-based modeling.
It would be the case, yes.
It will be about 50% of sequence and identity. Well, you can say that in some way it's an approach, right? It's an approach to the prediction of your model. But I think that as the same with the TM score, you have some accuracy scores also in this sequence modeling. So in this protein modeling, so it depends. I think that maybe you can approach to a close structure, a reliable structure. Also, if you use like these reliable predictors, and yeah.
I think also related to that question is even things that have less than 50% identity can be reliably detected as homologs. So as long as we find with some degree of certainty a good homology model, template-based approaches can be used. So in this example that we are doing right now, you'll see that actually the tool that performs the homology search is HH search. And you don't need a high sequence similarity to detect a homologue. So as long as you have a homologue, template-based approaches should work. Still, you do have to rely on these scores of certainty that Carolina was talking about.
Yeah, in this exercise specifically, this Swiss modeler they also have some accuracy scores, and you can compare between all your models predicted. In fact, we have added this useful site where you can read about this scores prediction. But the most used is called, in this case, is the GMQE. I will see how, okay. There's another question of Anindita Paul, is about, can you share your experiences with Rapido if you have used it in identification of functional modules within a protein?
Mm-hmm. So I have used this Rapido tool, but so what Rapido allows you to do is to superimpose structures, and it will find any instances where the algorithm has to break one of the structures to do a better superimposition. So it will work, for example, one of the ways you can use these results is if you have a multi-domain protein and one of them is in a different conformation, the algorithm will find the best answer for rigid bodies to be broken so that the superimposition is the best. I have used Rapido for multi-domain proteins that need to be compared when they have fold difference. But for the study of function specifically, because the way the questions I'm trying to ask with these tools are not related to function, but purely with the way proteins fold, I haven't used it for identification of functional sites. So yeah, maybe we can give you some references where you can find a better answer for that question. But Rapido is really a very good tool, in my opinion. So it works for identification of these instances.
Thank you, Claudia, for answering. So, okay. Oh, we missed this question from also, again, Clady Mirella, Osorio Mogollon. It is important the order of PDBS structure that I submit. But I think that was in a exercise.
The previous one, yeah. So the algorithm will take always these structure superimposition algorithms will always take one reference molecule and the one they are going to perform operations with. So you may have small differences, and the largest difference will be on the scores. TM-Align actually shows you the scores for each protein because usually you have proteins of different size. And because size is taken into account into all of these scores, that may make differences. Another thing is, because they always take one reference and one mobile structure, it is very good to identify for each algorithm which one will be used as a mobile one. Because if you want to use one structure as anchor, say for example, you have 7 structures of a homologue, and you want to then make a figure, for example, of all of these structures together. If you identify which one will move, you can just anchor your alignments to one of them and put all of them in the same space. So in general, yeah, there may be differences if you put one or another first.
Seems that we don't have any more questions for now, but we will wait for you to finish these 2 exercises, please. Okay, we're almost ready to finish this part of the tutorial. Very good.
Okay, we have just a few minutes to conclude this tutorial. It has been almost 3 hours, so this is the last part. Let me, as you finish this exercise, let me just share my screen one more time. And this last section is actually just wrapping up presentation. So we want to take many of the concepts we already described and discussed and try to make a little bit of sense of all of it. So this last part is called conservation and covariation. And the idea is to understand how change in sequence influences the change in structure. So at the beginning, we asked if we can study the evolution of molecules and how can we describe these relationships and the history of the molecules. And the answer is yes, we can take molecules and compare them and try to make a hypothesis of the evolution of these molecules using sequence. This is actually the way this phylogenetic tree was constructed, and then protein structure allowed us to visualize where the changes in sequence were happening. So this was not built the other way around, and maybe what we want is actually take a structure and then see what the evolution of this molecule was. So if we remember how molecular evolution is supposed to work, we would have a gene which has a DNA sequence, and then mutations at the gene level will sometimes translate into changes in the amino acid sequence. And I say sometimes because you know that, or you probably are aware of the generate property of the genetic code, which only means that there are more than one codons for a single amino acid in the genetic code. So some mutations will not have a clear translation into the amino acid sequence. And then when we have changes in the amino acid sequence, sometimes we will have a change that will produce, that we will have another amino acid, but with similar physicochemical properties. And if this happens, we would think that in protein structure, this change will not have a large effect, so that we will see an increasingly large effect as we go to the DNL level. What I mean by this is things that have a large effect in a mutation that has a lot of changes in the DNL level will probably not have so much impact on the sequence level and on the amino acid sequence level, and then changes in the amino acid sequence level not necessarily will produce a change in the structure. So this is a comparison of 346 homologs, and they are being compared in terms of sequence identity and RMSD. And for this set of homologs, we see that there is a relationship between the amount of change in sequence and the amount of change in structure. And we will think, great. So that means that if we see any change in structure, it can be directly related to changes in sequence, and we can reconstitute the history of these molecules using structure, because it would be a good probe of these changes that have happened. However, the paper I just showed you was written 20 years ago, and then now we have a lot more structures that have been determined, and also we have very good programs to study sequence similarity or to find more distantly related homologs. And if we compare these two plots, on the left we have proteins that are considered homologs, and on the right we have proteins that are structurally similar but are not considered homologs. And what we can see is that they have a very similar behavior. It's hard to tell if things that are very similar in structure, for example, everything that is below this, this bar would be very similar in structure, and still it doesn't look like they are homologs. So we have to be aware that we cannot rule out structural convergence, meaning that we cannot rule out that we have analogs in our set if we only use structural similarity. And actually, many of the proteins that share a fold are clustered in this twilight zone. What it means is that in this twilight zone, changes in the amino acid sequence start to have lost the record of evolution between proteins. So we cannot really say how far away or close are two proteins by looking at sequence. So this is the group of proteins that are going to be very difficult to study. So our hypothesis that we would have some kind of measurement of the evolutionary distance of two molecules just by looking at the structure doesn't seem to be so direct. It's possible that we can still extract some information, but it's not in a very direct path. Another thing that obscures our knowledge of the evolution, the closeness of two homologs, is something that happens at the tertiary structure level that will not be visible in the secondary structure level, which is, for example, this structure exchange. So sometimes in the three-dimensional structure, we see that a helix has interchanged for another, or a strand, in this case, has been interchanged. And this is only detectable on the three-dimensional structure, but the secondary structure will not tell us anything about this. Actually, the linearity of the sequence will be the same. But there are even other large rearrangements in sequence that will produce structures that are very similar. So all of these will confound, will blur our vision, and will make it very difficult to extract the evolutionary relationships between protein structures if we only use protein structure. The last thing to take into account is that, as Carolina already showed, proteins are not something that is fixed. They are not a rigid body as we want to think when we do the structure superimpositions, but proteins move. So they can have two conformations. And if we, for example, took these two structures and compared the RMSD or any other score, we would say, well, they are not very closely related if we only took RMSD as our indicator of closeness. So we have to be aware of these changes. Actually, if you do have something like that, it would be an easy way to detect this would be using a flexible structure superimposition method like these two, but there are many others, FATCAT and Rapido. And Rapido, I like it because it still treats your molecules as rigid bodies. It just makes cuts to improve the superimposition. And we go back to this slide. So we do know that protein structure can be a very good probe of distant relationships. And what we think is the point where we can extract the most information is taking structure and then informing the other levels on how should we analyze the evolution of our proteins. So for example, we can take a multiple protein superimposition, convert this superimposition into a sequence analysis, and then guide our multiple sequence alignment to have a better quality profile. And with this very high quality profile, we can study many properties like conservation, covariation, and see what it means in three-dimensional structure. So I'm sorry that the answer is there is no clear path on how to build a phylogeny from only protein structure, but we still can learn a lot from protein structure to infer evolution. This is the last presentation. We still have one small exercise that we can do in the next 10 minutes. It will be in the GitHub page as the other ones, and I just want to thank my fellowship and the lab where I work. And I am going to let Carolina also say goodbye, but we are still here, and we are going to wait until you finish your exercises, and we can communicate through the platform. So I don't know if you want to say some words, Carolina.
Yeah, of course. Thank you very much for being here. We will answer you again your questions in the platform. Don't hesitate us to send your doubts. Even in some days, don't worry, to us. And I want to acknowledge to my boss. We are in the Clara Chlohova Research Group in Charles University in Prague. So thank you very much for your presence, and please ask us everything that you want. And I hope you learned a lot. Yeah, thank you. Thank you very much. So we will continue doing the last section of exercises.
Yeah, so I'm going to share my screen. And this last exercise, so it's not very detailed, but it's actually the next step that will connect many of the analyses that we can do. So the rest of the analysis that we may want to do, like covariation or visualize change in sequence using structure, are things that we think would require to go very deep into the algorithms that are behind these calculations. So we probably, if you enjoyed this tutorial, we would probably think about doing another one where we would go deeper on what can we do after we found our homologous structures, what can we do with them? But yes, so this last one is going to be very useful for some of you, probably. We are going to take many protein structures, and we are going to construct a multiple sequence alignment. This Matraz server has the characteristic of not handling too many searches at the same time. It can get overwhelmed. I hope it doesn't happen to us this time, but if not, we have computed the results, and you can see the snapshots of what you would expect to have, and you can try it at a later time. But I hope you can do it. And then the last examples that I'm showing you here, yeah, for the steps on that, we would require another small tutorial. And yeah, we have around 5 more minutes. If you want to give us feedback on this tutorial, just let us know if you enjoyed it, if it was good, but we missed some interesting topics, let us know, drop us some lines in the chat. And you have, on the GitHub page, you have our email addresses. Let us know if you have something else to share with us.
Yeah, I think that everybody's finishing. Just as a reminder, if you want to really take into account the state-of-the-art predictors for disorder and also for modeling of structures, please consider to take a look into CASP in the case of disorder and order proteins, but in the prediction of specifically the disorder proteins, see the KIT site, please. Yeah, we will send you the links of these sites in the GitHub. They are already there, I think so. But thank you very much again.
Yeah, we really hope you enjoyed the tutorial. We really are very excited to share with you all of this. And yeah, let's try to make it a year-based tutorial. Maybe one day we will meet in person.